The core objective of genetic testing is precise: to identify changes in chromosomes, genes, or proteins to confirm a diagnosis, rule one out, or determine the statistical chance of passing on a specific disorder. But the physical scale of these disorders varies. An anomaly could be a massive whole chromosome error, like the extra copy of chromosome 21 that causes Down syndrome, or it could be a microscopic single gene mutation, like the specific error that causes sickle cell anemia. Standard imaging techniques like ultrasound are vital for assessing the overall physical shape and viability of a fetus, but they are blind to these microscopic chemical errors hiding inside the cells. This creates a unique challenge for clinicians. Because the errors occur at such vastly different sizes, there is no single universal genetic test that can catch everything. Effective diagnosis requires a tiered approach. A clinician must match a patient's clinical risk factors to the correct physical sampling technique, and then choose a laboratory assay with the exact resolution limits needed to see the suspected error. The clinical pathway usually begins with frontline, non-invasive techniques. This ultrasound scan shows the gross morphological details of a fetus. It allows doctors to determine fetal age, check viability, and look for major structural problems like cardiac defects or neural tube defects. Clinicians pair this with maternal serum screening, testing the mother's blood to analyze the concentration of alpha-fetoprotein, or AFP, a fetal glycoprotein. High AFP levels in the maternal bloodstream provide a secondary check for open neural tube defects. When non-invasive screens detect anomalies, or when demographic risk factors are present, the clinical pathway shifts. Doctors move to invasive sampling techniques to secure actual fetal DNA for direct analysis. This chart tracks the incidence of Down syndrome in liveborns based on maternal age. The risk curve accelerates sharply, jumping from 1 in 1,400 at age 20 to 1 in 100 by age 40. To acquire that DNA early, one option is chorionic villus sampling, or CVS. A doctor takes a transcervical or transabdominal biopsy of the placental tissue. CVS can be performed between the 10th and 12th weeks of pregnancy, yielding first trimester results. The alternative is amniocentesis. This involves the aspiration of the amniotic fluid surrounding the fetus. It is typically performed slightly later, around the 16th week of pregnancy. For high-risk cases utilizing in vitro fertilization, clinicians can bypass pregnancy entirely with pre-implantation genetic diagnosis. They extract a single cell from a 6 to 8 cell embryo before implantation to screen for specific genetic changes. Securing these physical samples through CVS and amniocentesis carries an inherent risk of pregnancy loss. Therefore, the decision to test is a strict mathematical calculus. Doctors only proceed when the statistical probability of a genetic anomaly outweighs the hazard of the procedure itself. Once the clinic successfully extracts a physical DNA sample, the laboratory takes over. The first analytical lens applied is typically classical cytogenetics. This is a karyotype. It is the traditional method of visualizing entire, intact chromosomes lined up in pairs, allowing lab technicians to manually scan for massive structural errors. This diagram maps the size of detectable genomic changes. Karyotyping operates at the macro level, limited to variations of 10 megabases and larger. At this resolution, it catches major structural errors, missing chromosomes, extra sets like triploidy, and balanced translocations where chromosome chunks swap. But this macroscopic view has a severe blind spot. It cannot resolve smaller chromosomal aberrations or tiny variants of unknown significance. Karyotyping is a wide net with massive holes. It proves the macrostructure of the genome is intact, but it says nothing about the millions of individual data points nested within it. To detect microdeletions or duplications, geneticists narrow their focal length using molecular cytogenetics, specifically techniques like fluorescent in situ hybridization, or FISH, and array CGA. Looking back at our chart, microarray technologies bridge the physical gap between whole chromosomes and single genes, extending detection capabilities down from 10 megabases to a 10 kilobase resolution. At this tighter resolution, array CGH effectively detects unbalanced translocations and microscopic variants that karyotyping cannot see. This scatter plot is an array CGH output. By zooming in, the technology loses the macro context. Because we are no longer looking at the physical shape of whole chromosomes, array CGH cannot detect balanced translocations or triploidy. Increasing the optical resolution forces a compromise. Clinicians gain the ability to see highly specific molecular details, but they lose the overarching architecture of the chromosome. Finally, we reach the smallest possible tier of genetic errors, the single gene mutations responsible for conditions like cystic fibrosis. Molecular genetic tests isolate extremely short lengths of DNA to identify individual nucleotide variations, finding a single miswritten chemical letter. On the resolution scale, genetic sequencing occupies the extreme right, drilling down to the level of one single base pair. At this absolute limit of magnification, we are no longer observing biological structures. We are reading the raw chemical code, letter by sequential letter. But because sequencing is so narrow in its focus, it is inefficient for diagnosing large-scale chromosomal duplications. Discovering a single base typo requires maximum microscopic resolution, which disqualifies macroscopic structural tests from diagnosing single gene disorders. Genetic diagnosis follows a workflow of escalating analytical precision. Categories of genetic disorders map to the tools required to see them. Broad chromosomal issues require wide lenses left, while single gene anomalies demand maximum sequencing right. Newborn screening identifies disorders like phenylketonuria early, while diagnostic testing isolates chemical causes behind specific physical symptoms. Similarly, carrier testing applies molecular screening to healthy individuals. It identifies if they hold single gene mutations linked to family history or ethnic ancestry. Every tier of testing involves the same compromise: width versus depth. A normal result on any of these tests does not provide a clean bill of genetic health. It only proves the absence of anomalies at that specific focal length. Precision medicine relies on a clinician's ability to deduce the physical size of the genetic error and deploy the exact right lens to find it.